Hi class, this is group HFF-1 and the experimental method we investigated was co-immunoprecipitation. Co-immunoprecipitation provides an in vivo experimental technique to show that two proteins are interacting via a protein complex bind. It is done by using target protein-specific antibodies to indirectly capture proteins that are bound to a specific target protein. Understanding and identifying specific protein-protein interactions are critical to understanding cellular functions, signaling, cell growth, and cell-to-cell interactions. In cell biology, the identification of a specific protein-protein interaction can lead to a better understanding of the cell's function as a whole.
First off, one must obtain their cell lysate in a container suspended in a cell lysis buffer solution. You will need to perform a wash in cold PBS before suspending the cells in your lysis buffer. In our hypothetical case, we are looking at proteins A and B in solution. We want to know whether or not protein B is binding to A. So in order to do this, we will add beads, often agarose or magnetic beads are used, which are coupled with antibodies specific to protein A, otherwise known as anti-A antibodies. The antibody must not bind in a region that would be affected by the binding of protein B to A, and thus is highly specific to an A epitope in a non-affected region. Monoclonal antibodies can be used and are incubated with the solution. Depending on what one is working with, it may take as little as 30 minutes or overnight for effective incubation. The solution is spun down in a centrifuge, and the pellet at the bottom will contain both proteins bound to the antibody protruding from the bead that has been precipitated out from solution if they bound. Anything not bound to protein A will remain in the supernatant and thus be discarded. The beads are washed away and the proteins are eluted out. There are a few different buffer solutions that can be used for this step, each coming with its own advantages. This step allows for the separation of the proteins from one another and antibodies. At this point, an STS page or Western blot must be performed in order to determine whether protein B is still present and thus bound to A. Western blotting allows for the identification of protein mixtures by molecular weight. Bands occurring for both proteins indicate that a co-immunoprecipitation as opposed to a simple precipitation has occurred. This next slide is just for your benefit if you want to look over the basic steps of this procedure.
So now we have a creative metaphor for co-immunoprecipitation that should hopefully help. If you've ever been crabbing before, you know what a crab pot looks like. We have some visuals attached here on the side. So fish can pass in and out through the little cage, but a little door on the side of the crab pot allows only crabs of a certain size to walk in but not walk out. When you bring up the crab pot from the sea floor, the only thing that should be in the cage are crabs. Now imagine there was a specific barnacle you were looking to study that only grew on Dungeness crabs. To find this barnacle, you would cast a pot to the bottom of the sea and wait for a crab to walk in. When you pull the pot up, all the other fish and sea life that were inhabiting the cage will fall through the holes on the side except for the crab because they are too large. Once the crab pot is brought back up, you take out the crab and run over its shell with your fingers to find any barnacles that are possibly attached. You know, any other sea barnacles not attached to the crab had to have been washed out of the cage. Only sea barnacles attached to the crab will appear, and the cage has to trap crab first and be brought up to the surface in order for you to see the barnacles. So on the next slide, we have a key for the metaphor that compares it to our methods. So we can see the crab pot would be our antibody, the crab would be our protein A, the barnacle would be our protein B. When we pull the crab pot out of the water, that can be seen as centrifuging the mixture and washing off other proteins that wouldn't stick to the antibody. And finally, using our fingers to run over the crab shell for barnacles is the same as using gel electrophoresis to detect protein B presence.
The article we chose uses co-immunoprecipitation techniques to analyze the HRAS pathway in mice. So from the article, we pulled the question, how can protein-protein interactions of the HRAS pathway be visualized? And next, what distinguishes cancerous HRAS state from the normal state? This led to the hypothesis that single molecule co-immunoprecipitation can differentiate cancer-specific HRAS signaling dynamics and determine the kinetics of intermediate and final complexes. The visual below is a description of how the cancerous cells were transformed into mice. Okay. The methods were broken down into 6 parts. The first one was gene construction and cell transfection, where the cDNA encoding HRAS was inserted either with mCherry or eGFP to generate plasmids. And these mCherry and eGFP are the fluorescent labels that can then allow it to be visualized. So for this, translocation to the plasma membrane was prevented. The binding domain was manipulated to generate expression, which is how it was fluorescently labeled. And then plasmid DNA was introduced into the cells for transfection. Next was the live cell colocalization study, which induced translocation to the membrane. So each HRAS was manipulated independently in order to isolate one on the membrane, and then multicolor and time-lapse imaging were used to analyze. Next was cell and tissue lysates preparation, just to get a baseline visualization in order to compare the manipulated pathways. The next part, which is also described in figure F from the article, was real-time single-molecule co-immunoprecipitation imaging, which is how HRAS was actually visualized. So for this, antibodies targeting labels were immobilized on the slide to pull down proteins. Cell extracts were then applied to the antibody immobilized surface and incubated. And then there was a buffer exchange with PBS to remove unwanted proteins, which then the slide could be visualized under a microscope. Next, to answer the second part of the original question, was transformation to cancerous cells. And so in this one, it compared the control inactive HRAS to oncogenic HRAS. Finally, was single molecule Western blot analysis, which is in figure C of the article, was used to confirm the findings.
As previously mentioned, co-immunoprecipitation was used to quantify the exchange selectivity of nature RAS proteins in xenograft tumors, normal tissue, and 2 cancer cell lines. RAS-RAF protein-protein interactions were analyzed using kinetic rates. Hong-Won Lee and the other scientists were then able to qualitatively characterize signaling proteins in tumor cells. As you can see in figure 3C, signaling proteins demonstrate that oncogenic RAS mutations selectively increase the active RAS fraction or the exchange selectivity between proteins by 1 order of magnitude. The most important aspect is that this occurs without affecting total RAS levels or significantly altering single molecule signaling kinetics. Furthermore, the RAS protein concentration can be increased when surface densities also increase. The application of single-molecule co-immunoprecipitation allows for characterization of protein complexes, such as quantifying the real fraction of active GTP-loaded RAS proteins. Scientists also observed that oncogenic mutations on the RAS protein take advantage of the collective behavior of these proteins and use it to increase in number. Although the pathway supporting these results is not yet clear, it is important to note co-immunoprecipitation has many applications. Specific antibodies and fluorescent tagging can be applied not only in cancer biology, but also as means of increasing precision and molecular diagnostics. It can also be used to study weak protein-protein interactions in more detail than ever before. We hope you enjoyed our presentation. Here are our citations, and thank you for listening.